practice now to be thinking in a detailed way, where the stakes are low. I know you think they're high, but they're not that high compared to someone who's sick and your health. Right? Any questions on the quiz? So these should be posted on Canvas probably later today, unless nothing goes calling it home. Hopefully not. So it should be on Canvas. All right. So I'm going to pass out the roll sheet. I almost scripted that. That would be really spectacular. I'm less likely to crash. All right. Oh, yeah. I'll make sure it gets back to you when it gets around. Okay. So I'm going to turn this. So last time we were together, we were reviewing some biochemistry, yes? You guys were super excited about it, as I recall. Enthusiasm, palpable. We're going to wrap up that review, and I just want to remind you that my goal in doing this review is not to teach you the biochemistry because you should have learned that in chemistry, but just to sort of jog your memory so that when we talk about these molecules, as we're about to do when we think about prokaryotic cell structure, that you remember what they are, okay? So we talked about carbohydrates last time. We said there's monosaccharides like glucose and fructose. There's disaccharides like sucrose and maltose and lactose. And then we said there are big polysaccharides like cellulose, glycogen, starch. We talked about all of those. The next, we're going to talk about 3 of the 4 big categories in this section. We're going to talk about nucleic acids when we talk about genetics. So the other 2 that we just want to review quickly is lipids and proteins. Lipids are a major component of cell membranes. They consist primarily of carbon, hydrogen, and oxygen. But the big characteristic that differentiates lipids from other biological molecules is that they are non-polar and insoluble in water. So if you wanted to take a bunch of cells and get all the lipids out of them, what you do is you put those cells in a blender and add an organic solvent. And anything that went into the oil layer instead of the water layer would end up being a lipid. So we think about several different kinds of lipids, and they have really different roles in cells and the human body as well. So there are simple lipids like triglycerides that are used to store energy. There are more complex lipids like phospholipids, sphingomyelin. These are really critical for forming the cell membrane. So every single one of your cells and bacterial cell has a lipid bilayer around it. It really acts like a little layer. In some ways, it's like a little layer of Vaseline holding the liquid into the cell, or aqueous material in the cell. And then there are steroids. So things like cholesterol is a steroid. There are steroid hormones. Bacteria don't have these, at least not the way we do. Things like estrogen and testosterone and progesterone are all steroids. Cholesterol is actually a really important part of cell membranes, by the way. This is a triglyceride. Again, just jogging your memory. Triglycerides have a glycerol molecule and 3 fatty acids, so it's a triglyceride, yes? And one of the things that you probably heard people talk about with respect to triglycerides is saturated and unsaturated, vegetable oil versus fat. Typically, we think about saturated fats, things that are like butter or lard. They tend to be solid at room temperature. They will have 3 saturated fatty acids where every carbon has the maximum number of hydrogens. An unsaturated fat has double bonds between the carbons, and so there actually, this carbon only has 1 hydrogen in this one, where the other ones have 2. So we say it's unsaturated because you can actually you can add hydrogens back by hydrogenating the fat. And so this is a unsaturated fat. If it has many double bonds, it's a polyunsaturated fat. So that's many, poly means many, right? And these tend to be healthier for you because these tend to be liquid at room temperature. And it actually has to do with the bend that you see here. Makes it harder to line up and form van der Waals interactions with other fats. And so because of that, the interactions between fatty acids aren't as strong. It melts at a lower temperature. And so it's a liquid at room temperature instead of a solid. All right, triglycerides have a primary role in the cell, which is energy storage. So and actually I really stated that poorly. We think of triglycerides as an organism more than a cell. So you have cells in your body devoted to storing triglycerides, and that's adipose or fat tissue, right? Now, phospholipids are are the lipids that are dedicated to forming cell membranes. So these are a little bit more complicated. They have, so here's our glycerol molecule, and they have two fatty acids, so it's not a triglyceride, but it's got two fatty acids. Usually one of them is unsaturated and the other one's saturated. And then on the third carbon, where you would normally have a fatty acid in a triglyceride, you have a phosphate group. It's actually more complicated, but it has phosphate and it has some other structures to it. And this ends up being very charged. This is a charged molecule. And so because it's charged, it's going to interact really well with water. Right? The fatty acids are greasy. They interact with oil. So you have this molecule that can't make up its mind who it is, right? It's got a polar end, likes to be with water. It's got a non-polar end that doesn't want to be around water. You know, if you shake up a bottle of oil and vinegar, the oil goes to the top and the water goes to the bottom, right? So this is a molecule that's like half oil, half water. And that's how it behaves. So if you look at these phospholipids, what they do when you put them together is they line up so all the oily parts are together, and then the charged parts will interact with water. And they form this really lovely bilayer, where you have phospholipids outside the cell, facing the outside where it's aqueous, where there's water. You have a bilayer of these greasy fatty acids forming basically an oily layer in the middle. And then inside the cell, where there's water again, you have another layer of phospholipids. So all of these molecules are happy. They're interact like dissolves like, we say in chemistry. Aqueous molecules want to be with other water-loving molecules. Non-polar or greasy molecules want to be with other non-polar greasy molecules. And so that's what the cell membrane does. It really takes advantage of this. And what ends up being amazing about the cell membrane is once you have this greasy layer between the two aqueous layers, you actually form a fairly effective block. Most molecules are too big to just go right through this greasy layer. And so the cell uses the membrane to control what goes in and out. The plasma membrane is kind of like the bouncer at a bar. It says you can come in, but you can't, right? And it's got some tricks that we'll sort of explain later on how they work in terms of controlling flow of materials in and out of the cell. Questions on that? So those are phospholipids. This is just a cool picture. This is a bigger section of the cell membrane showing the lipids in the middle here, and then different phospholipids. And so the outside has a lot of phosphatidylcholine. The inside layer has a lot of phosphatidylethanolamine, phosphatidylserine. Sphingomyelin is another type of lipid that you see a lot in cell membranes. So it's not just one kind of phospholipid, it's really a variety of them. And then the other thing that's shown here, and that we'll talk more about later, is these proteins that are sort of decorated inside the cell membrane, and they're going to be really important in that bouncer capability of the membrane, deciding who goes in and out of the cell and regulating the traffic. Are you ready for proteins? Okay, so proteins are essential to pretty much every function in the cell. When we talk about DNA later on, we're going to think about DNA as just a blueprint. It's not 100% true, especially RNA, which can have some enzymatic functions, but for the most part, DNA is just the directions, like a book. It says how to do something. And it's the proteins that actually are doing stuff, right? So the DNA basically tells the cell which proteins to make and when to make them, and how to make them. So proteins are needed for essentially every cell function. They include enzymes that catalyze chemical reactions. They include transporter proteins we were just talking about that allow things to go through the cell membrane. If it's a bacterium with a flagella, the flagella are made of proteins. It's a structural component. And then many bacterial toxins that we'll talk about later on as we focus more on disease and disease mechanisms. A lot of those are proteins as well. So proteins I have a laundry list, I think, on this slide. So transporters, receptors, regulator molecules, enzymes, communicate cells that talk to each other with proteins. They allow cells to be modal. They are part of the structural component of cells. Proteins do everything, right? I can't think of something they don't do other than store information. Proteins are made of amino acids. Remember last time we were talking about monomers and polymers? Sound familiar? So in this case, the polymer is the protein. It's many amino acids put together. And the monomers are these amino acids. So here are 2 amino acids. Actually, this R group is showing, this is a generic term showing that every amino acid has a different R group. So this is tyrosine, and this is just showing that if you change this to an H, it becomes lysine. If you change it to a CH3, it becomes alanine. So whatever this R group is, tells you what amino acid it is. The amino group, this is called the alpha carbon with the hydrogen, the carboxylic acid group. This backbone is the same for all of the 20 amino acids that we see in living things. Amino acids come in different sort of flavors. There are 20 of them. Some of them are positively charged, so histidine, arginine, and lysine are positively charged. Aspartic acid and glutamic acid are negatively charged. Do you think these amino acids like being around water or hate water? They're charged. So they're gonna like water. They're gonna be polar, very polar, okay? These amino acids, isoleucine, tryptophan, proline, valine, look at phenylalanine, its R group is a benzene ring. Alanine has a CH3. What do you think of this group? It's gonna like water or oil? I hear some people saying oil, right? So these are greasy amino acids. They're gonna be happier in an oily, hydrophobic environment. And these are the flexible amino acids. Glutamine, glycine, serine, tyrosine. All of these have some polar and some non-polar characteristics. And oftentimes, depending on the amino acids around them, they can sort of be flexible in terms of what environment they exist in. So there are 20 amino acids that we see in proteins, in bacteria and humans as well. Now, if you go to a health food store, you might see something that says there's more amino acids because they're trying to sell you stuff, right? So many years ago at Thanksgiving dinner, my brother-in-law he was lifting weights at the time trying to get really muscular. He brought this bottle that he bought at the nutrition store for dinner, and he was showing it to me because I was studying to get my doctorate at the time, and I was studying proteins. And he brought it to the Thanksgiving dinner, and so he showed it to me. It was really great thing, and I started to laugh. I was looking at the ingredients. It said it had 22 essential amino acids. And I said, well, first of all, only 20. Secondly, only 8 or 9 of them are essential. Maybe it's 11. But the point is, it's not all 20. Essential means your body you have to get them from your diet. Otherwise, your body can make these amino acids. So this whole 22 essential amino acids thing, there's a lot wrong with this. And then I looked at the ingredients, and there were some things in there that were molecules with amino groups and acid groups, but they weren't like ones that make protein. They were just molecules with aminos, and they were not amino acids anyway. So I had a really good laugh at his expense. And he has never asked me again for nutrition advice. I hope he's not still buying that stuff. All right. How do we hook amino acids together? They're hooked together through something called, if you're a chemist, they call it an amide bond. In biology, we refer to it as a peptide bond. And so here's our amino acid, and if you take an acid group and you hook it into an amino group, it's a dehydration reaction. You lose a water molecule, and you form a bond between the carboxy and the nitrogen, carboxyl carbon and the nitrogen. All right, and so you can hook up something as little as, I think, oxytocin is like 8 amino acids. It has lots of functions in the body, and then some proteins have 1,000 amino acids. So it has as many amino acids as it needs to perform the functions that it does in the cell. So far, so good? All right. I showed you some pictures last time of carbohydrates that had lots of sugar molecules hooked together, right? And then we just kind of looked at cellulose and glycogen and starch and kind of showed they had different structures. Because proteins are so different, because depending on how you hook the amino acids together, you can get radically different proteins. We actually will think about protein structure in levels. So one of the levels of protein structure is the primary structure. So this is just the sequence of amino acids. So asparagine, glycine, phenylalanine, glutamine, glutamic acid, glutamine, alanine, arginine, aspartic acid, cysteine, leucine, isoleucine, tryptophan. You get the idea. That's just the order that they're in the cell. Yeah? Make sense? That's called the primary structure. Simply the order. You pick the amino acids and lay them out in a row. But amino acids they don't form just strings. What they do once the cell starts to build them is they begin to fold. And you might imagine that amino acids with charged structures like this stick together, right? They're both polar. And non-polar amino acids, they like to be with each other. So as the protein is built, it begins to fold on itself to form what is the lowest energy structure for that protein. We also know now that there are molecules in the cell that assist in protein folding, and they're called, scientists are cute, chaperonins. They chaperone, right, the folding of the protein so that it folds in the correct orientation. So here's our primary structure. As the structure begins to fold, you can see it folding into, this is called an alpha helix. Sometimes the structure folds into kind of a sheet-like structure like this. So that's what they're trying to show here. It's kind of a flat, more sheet-like structure. Sometimes it's random, so they call this random coil, where it doesn't form a specific secondary structure. And then these structures, that's the secondary structure. So these are like sort of miniature structures. It's the second line of folding that happens, secondary structures. So things like the alpha helix and the beta pleated sheet or random coil would be secondary structures. Then these secondary structures will fold onto themselves. And so that's called tertiary structure. So do you see here that we have a tertiary structure? We have some random coil, some sheet, some alpha helix, and they are sort of together in a three-dimensional. This is actually a globular protein. This is showing one of the subunits of hemoglobin. If there's only one sequence of amino acids, it ends at tertiary structure. Sometimes there will be multiple strings of protein, and they come together to form something called a quaternary structure. So in hemoglobin, for example, there are two alpha subunits and two beta subunits. So there's actually 4 different proteins all come together in a quaternary structure to form the active hemoglobin molecule. And then inside that is an iron, a heme structure that holds the oxygen. It's a pretty complicated structure. All right, so primary, sequence of amino acids. Secondary, this is the secondary folding, things like an alpha helix, a beta sheet, random coil. Tertiary structure is a single chain of amino acids folded up. All right, and then quaternary structure is when you have multiple chains of amino acids that all come together to form a single active molecule. So those are the 4 different levels of protein structure. I have another picture of the same thing here, just because sometimes different pictures speak to people differently. Here's our primary structure. Here's our secondary structure. This is an alpha helix. Here's a beta sheet. And then these are going to fold up into a tertiary structure. So you see the tertiary structure here. We've got some sheet. We've got some alpha helix. We have some random coil. All right, and then if you get 3 or 4 of these hooked together, this is showing 2. You can see that there's 2 different strands of amino acids. That would be your quaternary structure. One nice thing that this slide shows that didn't show on the other slide is just the interactions between the amino acids. So these 2 amino acids aren't next to each other in the chain, but you see how they're interacting with a positive and negative charge? So that's holding that part of that loop together. Even though those amino acids are next to each other, they're being held together at a distance from that electrostatic attraction that they have, the positive and negative charges. This is actually a covalent bond. So between cysteine, you can actually hold a protein together with a covalent bond called a disulfide bridge. And this is really important in a molecule that hopefully you're all making and don't have to inject, which is called insulin. So insulin actually is held together with disulfide bridges. Here's a hydrogen bonding. This is a hydrophobic interaction. So you can have 2 amino acids on totally different ends of a primary sequence that are right next to each other and interacting. And they have lots of covalent bonds, ionic bonds, hydrogen bonds, van der Waals forces. All of those are ways that different amino acids can be held. Okay, that's going to give that protein its shape, and its shape is then going to dictate what it can do in the cell. Okay. All right, let's take a little eye clicker break. What kind of molecule is amino acid? All right, last chance to click in. All right, so still a little bit of a struggle with this, right? A monomer means 1, right? Monomer. Polymer So it would be a monomer. And then if the amino acid is a monomer, what would be the polymer it forms? The protein, where you're hooking multiple amino acids together, and you would have many amino acids to make a protein. Okay, so amino acid is a monomer, and a protein is going to be a polymer. This is just a cool picture. But I like it because it kind of illustrates what we've been talking about, right? So this is a tertiary structure, and you see the alpha helix and the beta sheets here. There's a random coil. All right, so this is a globular protein that's being held together. It's an enzyme, and I know that because it's got this ACE at the end. So anytime a protein has ACE at the end, it means some kind of enzyme. All right, so thinking about this picture now, do you see that this molecule has an exterior, but it also has an interior? Yeah? Okay, so now let's look at the study guide question, and we'll go back to this maybe to help us think about it. Oopsie, that's not the right version. So many proteins that are soluble in water have a hydrophobic interior, but an outside layer of hydrophilic amino acids. So let's say that, let's kind of look at this and think about it. So what we're saying is that the outside of this molecule, all the things that are touching the outside are polar. They like to be around water. But the inside of this molecule is hydrophobic. It doesn't like water. What's the advantage of having a hydrophobic interior if you want to be a hydrophilic protein that's soluble in water? What do you think that is? To keep it tight. And so I know what you mean, and you're right. What it's doing is it's holding the molecule together. If that protein starts to unfold, those hydrophobic amino acids don't want to be around water, right? So they're going to tend to stay together and hold the structure together. Does that make sense? So by being hydrophobic in the interior, it lends the molecule a lot of stability. Let me give you an example of this that you probably haven't thought of, but you do it all the time if you eat eggs. You ever fried an egg or hard-boiled an egg? So you know how when you first crack the egg before it's cooked, the egg white is white? So that albumin protein that's in the egg is soluble in water. That's why it's clear. It's all dissolved in an aqueous. It's kind of thick solution, but it's dissolved. And those molecules are just like this one that we just described. They're hydrophilic on the outside, but they're hydrophobic on the inside. Now, when you apply heat, eventually there's enough heat to unfold those molecules. So they unfold, but once they unfold, those hydrophobic amino acids in the center, they don't like to be around water. And so what they do is they fall out of solution and they turn opaque. Now your egg is turned bright white because you cooked it. You basically denatured or unfolded the proteins that are in the egg white. Right? Does that make sense? Okay, so you've exposed them. They were in the interior of the albumin, now they've unfolded and they've essentially precipitated out of solution. Okay. All right. Enzymes are a really important subcategory of proteins, and we're going to spend a lot of time talking about enzymes this semester because enzymes they catalyze the chemical reactions in the cell. Cell chemistry wouldn't occur without those chemical reactions. So in this case, we've got a picture of 3 different molecules. So caseins or proteins, this has lipids in it, and this has starch. I think we looked at this last time. But in all cases, the bacteria are actually making enzymes that secrete out of the cells, and that allows the bacteria to take up the molecules that are inside. Okay? So this is a way for bacteria to get amino acids, to get the products to build up lipids, and to get sugars into the cell. All right, this is a cool picture because this is actually, you don't panic. This is a DNA molecule, and this is an enzyme. And you can see the parts of the DNA molecule inside the enzyme. And the DNA molecule is actually repairing, or excuse me, the amino acid here. See the alpha helix, the random coil? It's actually repairing the DNA. That's what that's trying to show you there. Okay? So you can see how the protein is interacting, wrapping itself around the DNA molecule and doing a repair. You don't have to know this. It's just trying to visualize some of the amazing things that these proteins do. Okay, that's my last slide. That's my last review of macromolecules. I'm hoping that's enough so that you feel sort of refreshed and ready to talk about these molecules more from a cell structure point of view. So it's 11:53. 3, I was thinking we would do our disease of the day, and then we'd start talking about prokaryotic cell structure. Take a little break from chemistry here and think about a disease. All right, so the next disease we're going to talk about, with the exception of the plague, I think almost all of the diseases we talk about are things that happen all the time. So the plague is sort of not a huge, it does happen still, it's not a dead disease, but most of our diseases of the day are common things that you might actually really encounter as a healthcare practitioner. Hopefully not Ebola, although if you go work for Doctors Without Borders, you can easily interact with things like Ebola and certainly Zika in the United States. So with the exception of the plague, so Staph aureus is one of these garden variety pathogens that you probably see quite a bit of in the United States in a healthcare setting. And it causes a bunch of different problems, but the two really common ones are impetigo and abscess. Staph aureus is a common organism that's found in your nasal passages on your skin. About a third of people have Staph aureus in their microbiome. We swabbed you, we find it on about a third of people. Let me close this. Oh shoot. That makes it sound worse, huh? You know what?
I think it's from outside.
Yeah, it's a car.
I think it's next door in the. It's bothering me. Is it bothering you? Let me go. It's the alarm on the dishwasher. So let me go tell Nick to turn it off.
CO and double CUS.
The second, the aureus, is lowercase. That's the typo.
All right. I had a button that says silence alarm. Okay. So Staph aureus is a very common organism. It's found in lots of people's skin. It's not good or bad if you have it in your skin. It oftentimes will just live on the surface of your skin without causing any problems. It's we call it a halophile. It likes salt. So your skin's actually very salty as an environment. It's one of the ways your body controls the microbes on it, is to become a salty environment. But Staph likes salt. So it's happy on a very salty and fairly dry environment of your skin. One of the things Staph can cause is something called impetigo. This is a very non-lethal, non-big deal kind of infection. You want to treat it. It's possible that impetigo starts with Streptococcus pyogenes, which causes strep throat. But then Staph almost always takes over. So by the time the doctor's doing a culture, oftentimes all they see is Staph aureus. But it creates this kind of crusty, flaky lesion, very easy to control with antibiotics, but very contagious. So if it gets in a daycare center, for example, it'll sort of go from person to person very quickly. But the beauty of Staph aureus is it causes all sorts of skin and soft tissue kinds of infections. So another infection that Staph can cause is something called scalded skin syndrome. This is a lot more serious. We see staphylococcal scalded skin, that's a tongue twister. Staphylococcal scalded skin syndrome is commonly seen in babies. And it usually starts with a little infection at the umbilical cord, and then it will spread. The bacteria can get into the blood, or the toxins from the bacteria can get into the blood. And it's called an exfoliative toxin. So not all Staph has this toxin, but some does. Some strains of Staph have it. And the toxin causes the skin to detach near the dermis. And so what you see is this peel. It looks like a really bad sunburn as the skin is peeling from the effect of this toxin. Usually babies do okay. They get treated with antibiotics and they recover. Adults, though, this is often lethal. You tend to only see it in adults who are already immunocompromised, and so this a big skin infection on top of the other problems they have, oftentimes results in death, so you get a much higher mortality rate. The big concern with anything that's doing damage to the skin are secondary infections. So if this is the primary infection, the first infection, primary infections are when bacteria attack healthy people. Secondary infection would be some other organism that can then get into the skin and make you sick, because the skin is your first line of defense from pathogens. So if your skin's peeling off, then other organisms can get in there, right? All right, another thing that staph can cause is abscesses. Very famous for causing abscesses. Abscesses are when you get, oftentimes they result in a puncture wound. So basically something goes in under the skin and delivers bacteria under the skin. Like, for example, using IV drugs, really common way to get an abscess. In fact, that's what this is showing here, someone who's got an abscess on their arm. So basically they get a wound where they get bacteria inserted under the skin, the bacteria begin to divide, your immune system walls off the infection, and a bunch of neutrophils come to attack the bacteria. So you get this pus pocket full of bacteria and neutrophils, and your body is trying to control the infection by keeping it walled off in this abscess. So this is an opportunistic pathogen. This is a bacterium that's getting in and causing this infection because it had the opportunity. You know, you gave it the opportunity if you used a needle that wasn't clean. Another common kind of abscess is cats. They're outdoors, they get into fights, and they get puncture wounds from fighting with other cats. I had a cat once that, it seems like I was taking him to get abscesses drained. I don't know how many times I took him to get abscesses drained in his lifetime. It was more than 10. I got pretty good at draining them myself to save the vet bills. But oftentimes they would come back because you need to put a tube in and really drain them effectively. It was harder for me to do it. Anyway, cats are kind of famous for getting abscesses. That cat died. I should tell you about that cat. He died peacefully in the vet's office of old age. And I put him to sleep. And he should have died a violent death because he tried really hard. I've had other cats that were really sweet that didn't go so well for them at the end. But that cat died peacefully, and I was shocked that he made it to old age. I would find him like a city away. He'd wander. People would call me and say, "Your cat's in my yard." He was going to people's houses. One year for Halloween, I was living in Palo Alto with my brother. We had rented this is before Palo Alto was unaffordable. Well, we were already in a house that was falling down. And anyway, the kids were coming for Halloween, and we had candy. And every time, I had the cat in for the night because it was Halloween. And every time someone come to our house, they say, "Oh, Marty, he comes to our house all the time." Don't let him in your house. So cats get abscesses too. Puncture wounds cause abscesses. And it's from the staph that's already there in your skin getting under the wound. Another thing staph does, in case you didn't feel like I've given you enough diseases that are caused by staph, another one that it can cause is something called endocarditis. And so this is where the staph gets into the blood and actually coats the valves of the heart. All right?